Okay, so let's look at some immunohistochemical markers, right? We'll be discussing immunohistochemical markers and we'll be starting from the definition to some examples of what immunohistochemical markers, right? So to me, what I know about immunohistochemical markers is, okay, let me say, probably you are affected by a certain disease condition and there's a way they can actually take some samples from your body and get to like test these samples, and if there are some particular things or particular markers now that they see in this sample, that they get to actually conclude that, okay, this guy is yeah, it's concluded that, okay, 100% being infected by this disease condition. All right, so immunohistochemical markers. mm, you see, like if they take a sample from the pancreas, now they test it, right, and they get to see if there are some particular immune stuffs, okay, in that sample. Once they see it, then it's like confirmed that, okay, you have this disease condition. All right, so guys, so these immunohistochemical markers are used to identify specific proteins or antigens in tissue samples. All right, so they are essential in diagnostics pathology, particularly in cancer diagnosis and classification. Okay, so we're looking at some examples of immunohistochemical markers because that's like the main things that we are supposed to look at. All right, this lecture was made for us. This lecture was made just so we can actually look at some examples of what immunohistochemical markers. All right, so the first one is what? Cytokeratins. And these ones, they are used to identify epithelial cells and tumors, right? So we have example cytokeratin 7, cytokeratin 20. Then we have Vimentin. This one identifies what? Mesenchymal cells and tumors, right? Examples like tumors like sarcomas, okay? Then we have the CD450, CD45, sorry. CD45 identifies leukocytes and lymphomas. Then we have the CD3. This one identifies T cells and T cell lymphomas. Then CD20, it identify what? B cells and B cell lymphomas. All right. Then we have the P63. This one identifies basal or myoepithelial cells and tumors, right? Like breast cancer, prostate cancer, and the rest. Then we have the S100. This one identifies neural and melanocytic cells and tumors, right? Like melanoma. Then we have the HMB45. This one identifies melanocytic cells and tumors, like melanoma too. Then we have the chromogranin A. This one identifies what? Neuroendocrine cells and tumors, like examples like carcinoid. Then we have the synaptophysin. This one identifies what? Neuroendocrine cells and what? Tumors too. Then we have the Ki67, also known as MIB1. This one identifies what? Proliferating cells and tumors, right? So this is like immunohistochemical assay. All right, they do it purposely once they are able to see some particular immune markers, right? Then you know that, okay, these immune markers were formed or released because of a particular disease condition in the body. We have the P53. This one identified tumors with mutated what? P53. Then we have the ER, that's the estrogen receptor, and PR, uh, progesterone receptor. This one identify immune receptor positive tumors like breast cancer. Then we have the HER2 or the NEU. This one identify HER2 positive tumors like breast cancer. Then followed by that, we have the PDL1. The PDL1 identifies what? Tumors with potential response to immunotherapy. I would say that these markers help pathologists to classify tumors, determine their origin, and predict their behavior. All right. The choice of immunohistochemical markers depends on the specific tumor type or diagnostic question in view. Okay, it basically depends on what you are trying to actually look out for. Okay, but this immunohistochemical assay, right, how to do the test is basically one way. Okay, and anything you see should actually determine uh, what is underlying. Okay, so that's it about this immunohistochemical markers, talking about examples and uses. Okay, so that's it and bye.